Thank you, everyone, for being here and for waiting these few minutes before to allow everyone to step in if they are interested in this webinar. This is and welcome to this new webinar of by the Global Plant Council. This is the first chapter of a webinar series that we have called Plant Science-Based Solutions to Tackle Climate Change that we have organized leading up to the Climate Summit that will happen by the end of this month in Dubai. And today's webinar, we have some novelties. Today's webinar will be moderated by Liana Acevedo-Siaca, who is a board member of the Global Plant Council and a researcher at the Michigan State University. And I will stay behind the scenes, moderating the chat and in checking your questions, your answers, and answer them if appropriate. So, Liana, I give you the floor.
Good morning, good afternoon, and good evening to everyone. Thank you so much for joining. Very quickly, some housekeeping as well. This session will be recorded, just so everyone is aware. And to reiterate what Isabel had mentioned, we want to thank you so much for joining us for our first webinar, which is Unveiling the Power of Plant Science. This is, as was mentioned, our first webinar of a larger series of webinars called Plant Science-Based Solutions to Climate Change, and a little bit of background on the Global Plant Council for those who might not be who might want to know a little bit more about what we do. The Global Plant Council was founded in 2009 to provide a body that can speak on behalf of the plant science community with a single strong voice in the policy and decision-making arena, and to promote plant science research and teaching around the world. So this works towards that mission. If you're interested and you'd like to learn more about the Global Plant Council and our different activities and initiatives, please feel free to visit the Global Plant Council website, especially the one on webinars, which I'll share in the chat box shortly. So for today we'll be hearing two different talks from panelists Dr. Claudia Stange and Dr. Rodrigo Gutierrez. Each speaker will present roughly a 20-minute talk on their work. Also a little bit of housekeeping. Please submit any questions through the Q&A box that's provided through Zoom, as we will begin the question and answer portion of the session at the end after both of the talks are complete. Questions can be general or directed to specific panelists, but that's where we will manage everything. And so now let's get directly to the science. I'm very pleased to introduce our first speaker, Dr. Claudia Stange. She is a Chilean biochemist and plant molecular biologist who is currently serving as a full professor at the Universidad de Chile. Her research spans from micropropagation of radiata pine to groundbreaking work on carotenoids and carrots. And today she will shed some light on genetic plant improvement applied to climate change. So with that, please, Claudia, if you can take the stage and give us your presentation.
Thank you, Liana, for your presentation. And it's okay. Are you seeing the presentation or not?
Yeah, I think you just have to go into presenter mode and then we should be good to go.
Why is this happening? Yeah, a ver. Yeah. Now
looks great. Yep, looks fantastic.
Okay. Yeah. Good afternoon, good evening, everyone. It's a pleasure for me being here, and thank you for the invitation. Let's start. So as we well know, the climate crisis is putting the planet's agriculture in risk. And we already don't know how to feed around 9 billion people by 2050. So it's a big problem because the arable land for food, feed, and fiber production is very less, no? It's around 10% of all the land in the earth. And why is this problem so big? Because the climate crisis is producing a lot of land unable to use as arable land, because this 40%, around 40% of the Earth's surface is affected by drought and salinity. As you may see here in the picture, all the land that is depicted in red or orange are affected severely by drought. And the climate crisis also affects the biodiversity and produce soil erosion. So what happens with the plants? Plants also are affected, of course, no, because they grow less, slower, and produce less fruits. Indeed, the crop productivity could decrease 50% due to the climate change, specifically due to the drought and salinity of the soils in which these plants are grown. And how we can contribute from the science and technology to face with this problems and to achieve a sustainable agriculture. So with this in mind, we have developed several strategies. One of them is to the formation of the APSA Chile committee, and also through several projects that are focused in producing new plant varieties aimed to face with this climate change or climate crisis. Regarding the APSA, who we are? We are a people of scientists, academics, entrepreneurs who share the vision that plant biotechnology is a powerful tool for genetic improvement to contribute to agri-food sustainability. What are we doing? So we meet with politicians, we develop seminars, several surveys on plant biotechnology, and so on. And indeed, in several weeks more, we are organizing a seminar entitled The Importance of Plant Biotechnology in Genetic Improvement to contribute to food safety and sustainability. And it will be held on 7 December in a hybrid mode if you want to join us. But today I will share with you our strategies about the project that is focused in increasing plant tolerance to the effect of climate change. This is a project that started at the beginning of the pandemic and finished 2 months ago. The project that was funded by national grants had 3 aims, but today I will focus this presentation on the first aim, that is regarding the development of kiwi and tomato rootstocks more tolerant to drought and salinity using gene edition. Why tomatoes? Because tomato is the most consumed and produced vegetable in the world and the most exported by Chile. But the project is focused in an endemic variety named Poncho Negro that is grown in the north of Chile, that is not under intellectual property protection. And our focus is to give value to an endemic variety by creating rootstocks that are more tolerant to drought and salinity. In the case of kiwi, we are working with this fruit because it is very important for the Chilean economy, but it is very sensitive to salinity. Specifically, we are working with the Highward variety, this commercial variety that produces the green fruits. But this variety is also used as a rootstock. So we want to generate new rootstocks that could be more tolerant to raw and salinity. So the big question to start this project was which genes could be good targets to be edited in these two plants. For this, we have to know and to remember the mechanism that plants have to overcome the abiotic stress of the soil, of the environment. So once plant sense the osmotic or the ionic stress, they activate several mechanisms in which ABA, the abscisic acid hormone, it could be or could be not involved. So once it is activated, several protein kinases, transcription factors are activated. So given to the expression of genes that are involved in stress tolerance. But together with these positive regulators that we name them, no, because they promote the abiotic stress tolerance, there are also other factors that we name them as negative regulators because they impair the effect of the positive regulators. So the role of these factors are to maintain the homeostasis once the effect, the abiotic stress is gone. But if these negative regulators are overexpressed, we are able to see that the plants are more higher sensitive to these abiotic stresses. So our hypothesis is that when we edit the negative regulator genes, we're able to obtain a higher response and tolerance to raw and salinity because the positive regulators are more active, are more active. So then we, with this in mind, we look for negative regulators genes in papers, in transcriptomic databases, and also in RNA-seq that have been done in plants under abiotic stress and so on. And we had a very huge list of genes that participate during these processes. But we selected in this short list only those genes that are expressed, that are significantly expressed during abiotic stress. And once they are overexpressed in plants, they produce sensitivity to draw or salinity, or when they are mutated, they produce more tolerance to the abiotic stress factors. So some of these genes code for enzymes that participate in the synthesis of ethylene, that is a well-known hormone that produces or participates during maturity and fruit ripening and senescence, but it impairs the tolerance of abiotic stress in plants. The other genes that we selected code for E3 ubiquitin ligases that are very specific enzymes that send several genes, several proteins, for degradation. In this case, there are several ubiquitin ligases like this one that sends these positive factors for degradation. So, and these factors participates in the draw stress response. So this is an overview of the role of the ubiquitin ligases as negative regulators in abiotic stress response. So once we selected these genes, we have to look for those genes in our genome, in the Kiwi and in the Poncho Negro genomes, searching for the corresponding orthologs. So for that aim, we have to sequence the genomes of Poncho Negro and Hayward. We did it by Illumina and PacBio, obtaining a completeness of 98 and 96%. That is very high, very good, when we compare it with the reference genome that is available online. So when we select those genes, we then perform several deep bioinformatic analysis of those orthologs, analyzing if they have the putative functional domains, we perform the phylogenetic trees, and so on. And then we select the list of genes and analyze the expression of those genes in RNA-seq that we also carried out between plants that were subjected to draw or salinity at 6 and for 24 hours. As you may see here, some of the genes are downregulated during this process, during the stress, but others are upregulated. And these kind of genes are of our interest. In parallel, we also performed a qPCR of some of the genes that we selected, noticing that some of them, like ACS6 that participate in the synthesis of ethylene, is induced only under salt stress, and sub7 that code for E3 ubiquitin ligases is induced under draw and salinity. So these two genes were selected for editing, and it is interesting to note that the expression profile of both genes by qPCR is more or less similar than the RNA-seq. So these genes were selected for Kiwi. In the case of Poncho Negro, we performed a qPCR of all the selected genes, and we observed that some of them were induced under drought and salinity. Yeah. I'm sorry, but I'm here. It's okay now?
Yes. See you here perfectly.
Okay. Yeah. So some of them are induced under salinity and raw, but other ones are not significantly induced. So about this analysis, we selected some of them to produce the vectors, the CRISPR vectors. And then once we have the vectors, we have to choose the strategy for transformation. So the main aim is to obtain non-GMO edited plants. So we selected the protocol that was described by Chen in 2019, in which the plants after the explants after they were transformed to Arboretum tumefaciens, they were selected, they were grown, they were regenerated, but without selection. So the medium has non-antibiotic for selection. So it's very different from the above strategy. So the edited plants have to be selected only by molecular and functional analysis. It's very hard and time consuming because we produce 3 times more plants than this strategy. So, but here we are. So we started the transformation of Kiwi following the protocol that was published by Luis Felipe in his doctoral thesis. And after 5 months, the explants produced the kaiju and induced the proliferation of the shoots. The kaiju were used for functional evaluation of the vectors by PCR, and these additional bands reflect that they are possibly producing some addition in the candidate genes. After 4 months, the plants that were regenerated were transferred to another medium, expecting they elongate. In the case of Poncho Negro, we expected it would be easier because it is a tomato plant, but it wasn't. It was very hard to standardize the in vitro regeneration and transformation of Poncho Negro. It took around 10 months until we have the regenerated kaiji and the plants after the transformation with the CRISPR vectors. Some of the kaiji were used for the vector evaluation, and the plants were then transferred for elongation. In this case, we also used a medium without antibiotic. Unfortunately, the Poncho Negro, several lines of Poncho Negro died during this process. We don't know why, but this plant is very sensitive. So, well, we lost several lines and only 2 of them left until now. Then we, regarding Kiwi, we have 32 regenerated seedlings. 9 of them were evaluated at the molecular level in order to know if they are transgenic for Cas or not. We are interested in those plants that are not transgenic, but we also include in our future analysis the transgenic lines for comparing. As you can see here, the PCR shows that the line 6 is transgenic. We always perform these PCRs in triplicate to be sure that the plant is or not transgenic. So, and in the case of Poncho Negro, both plants are not transgenic or are not transgenic, but we don't know if they are wild type or are non-GMO edited plants yet. So we are already sending the fragments of the genes for sequencing to know if they are edited or not. And in parallel, we are performing or we are starting with preliminary functional assay in the case of Kiwi to detect if some of the plants are more tolerant to salt stress than the wild-type plant, or the control plant, which is a transgenic line that overexpresses a transcription factor that is a positive regulator of salt stress. We also include the transgenic line as a control. So this is an ongoing assay because after 15 days, we are not able to see any difference in most of the lines evaluated. So what's next? As you can see, we are at the functional and the molecular analysis of the plants in the in vitro or in the greenhouse condition. And in this roadmap, we are half-life, and there is a lot of work left to do, including the grafting and field assay. So to finish, I would like to thank the team, especially to Samuel, to Christian, Leticia, Claudia, Max, and Jorge for the work that they did, and also for our funding, and of course for you for your attention. Thank you.
Thanks so much, Claudia, for your talk. I think it's also important to point out that even though your work is really focused on tomato and kiwi, which are huge agricultural and economic focus in Chile, that this really has so many applications to different crops around the world. I mean, the problems related to drought and salinity go far beyond borders in this case. So it was really interesting to hear your perspectives and your work on this. With that, we're going to move on to our second speaker. Just a kind reminder that we will be taking questions at the end of both of the talks. And that we ask audience members to please submit any questions into the Q&A box, and then we'll have a look at those. But now it's my pleasure to introduce our second speaker, Dr. Rodrigo Gutierrez. Dr. Rodrigo Gutierrez holds a PhD in biochemistry and molecular biology. He currently serves as a full professor at the Pontifical Catholic University of Chile. He leads the systems biology laboratory. His research is twofold: delving into the molecular intricacies of how plants perceive and respond to nitrogen nutrients, and also exploring the adaptive mechanisms of wild plant species in challenging conditions of the Atacama Desert. So with that, Rodrigo, if you'd like to start, we'd be really keen to hear your talk.
Yes, thank you very much for the opportunity to share some of our work. As you mentioned, one of our main interests is to try to understand how plants manage to survive in the extreme conditions of the Atacama Desert. So we will be moving to the wild in this presentation. As already Claudia introduced, we as human society are facing an ecological crisis of global proportions, primarily due to the accumulation of greenhouse gases in the atmosphere that is shifting the world's radiative balance and increasing global temperatures. And there is a lot of predictions, and actually some of experiences in Chile, for example, that this will lead to changes in climate patterns, and one of the effects will be increased global desertification. And this would be primarily affecting subtropical areas such as Chile, but any other country in those latitudes will suffer similarly. And of course, an increase in desertification poses serious challenges for plant growth, and this will have an impact in crop yield and therefore food security that Claudia also introduced, that it's not only that we will have more people, but also the conditions will be more challenging to secure food production. So if we think about ways to tackle the problem of facing increased desertification, some years ago, we started to look into our local ecosystems. And we realized that the Atacama Desert is really a unique earth system. And it's probably one of the harshest environments for life in general, and also for plant life. It's a desert that's between the Andes Mountains and the Pacific Ocean in South America, and is thought to be the driest and also probably the oldest non-polar desert on Earth. It has experienced semi-arid conditions probably for the past 150 million years. And there's some various estimates that indicate the desert inner core has been hyperarid for roughly 10 to 15 million years ago. And this is where we are focusing on the work that I will telling you about. And this is probably due to a combination of factors that, if anybody's interested, we can discuss later on. But in addition to obviously being a desert, and therefore life has to endure extreme low water availability, there are other conditions that are not very friendly. One of them is extreme daily temperature oscillations, high salinity in the soil, and several parts of the desert. It's also one of the places on Earth that receives the highest radiation. And in addition to that, the soils are very poor. So as I mentioned in the title of my presentation, if you saw it, this is not an extreme environment, but also there's several extremes that life has to survive in order to thrive and succeed. Despite all these challenges, if you know where to look, there is surprising plant diversity that caught my attention the first time that I went there more than a decade ago. And this is, of course, not in oases where there is water availability, but out in the field. And not only there is a surprising plant diversity for which we know very little about. Most of these species have never been studied. We have the botanical names and botanical descriptions, but we know very little about them. And some of them are relative to plants of great interest, such as, for example, the one that's shown here, pictured here, Solanum chilense, which is a relative of tomato, one of the species that Claudia was talking about. There is some lupin species as well, which are relatives to legumes, and lupins that are already cultivated. And so on, several grasses. And so we think there's great potential in studying this surprising plant diversity that is able to survive in the extreme Atacama Desert, to learn about mechanisms that then can be translated to crops. I should also say, and I won't have time to talk about this in this presentation, but some of these species have intrinsic value because they were used by local cultures as food or sources of, for example, drugs for health issues that we're also exploring. And so not only we can extrapolate, but some of these species can have value on themselves. So I think it's very interesting to study these wild species. So we have been concentrated in an altitudinal transect that's shown in this picture, just to orient the audience. So we are here located in South America. Our study site is just off the Atacama Salt Lake, and it's an altitudinal transect that spans the limit for macroscopic life in the desert. Below this point, which is marked here as the limit of absolute desert, and all the way to the coast of the Pacific Ocean, there is no more macroscopic life because there is not enough water. But if after this point, as you go up in the mountains, this is about 2,400 meters above sea level, there is enough humidity and water availability that supports plant life and also animal life up to about 4,600 meters above sea level, where above that it's just too cold. So you have these two limits, limits of absolute desert, limit for life in altitude. And between these two extremes, we have selected 22 locations where we have been going back since 2010 to study diversity and other aspects. Some of them I will mention today. And this project has attracted several colleagues from around the world to collaborate with us to characterize multiple aspects. And I'll be touching about some of the work that we have done with Gloria Carusi at New York University, and also some of our French colleagues where we have done some metabolomics work of these species. So here I have a short video illustrating some of the work that we do. So we took everything that we need to the field, like liquid nitrogen, to collect samples, and we collect the plant tissues, do treatments as necessary for the goals of our projects. It's not necessarily easy working at 4,400 meters above sea level, but I hope to entice some of you that, to come and join us on some of the projects that we are developing in Chile. Because this is a short presentation, I don't have too much time to go into the details. So I will just say that we have measured environmental parameters for many years now. We have meteorological stations. We have done detailed analysis of the soil substrate as well. And we have found, I think, three main factors that shape the ecosystems that are found in this altitudinal transect, which is water availability, as I already mentioned, temperature, but also the lack of nitrogen and radiation. So the highest ecosystem we can find is called the steppe, and it's dominated by grasses, perennial grasses, which is over 4,000 meters above sea level. Then we have the puna, which is the richest ecosystem, which is dominated by shrubs and subshrubs, and the prepuna, which is the most arid ecosystem, which is dominated by annual plants and succulents. Studying diversity in an extreme environment is not so easy. We spent several years just trying to characterize the diversity of these three ecosystems along the mountain. And this is summarized here. We identify 93 taxa using a combination of botanical surveys, soil DNA analysis using metagenomic sequencing, pollen analysis, soil seed bank, and so on. And out of these, we could identify 78 plant species in these three ecosystems. Beyond going into the details, I think it's very important to understand what the true biodiversity is in a given ecosystem, because this gives you sort of a baseline on what currently exists. And we can do two things. So we can go back in the past and see how this ecosystem changed over time. And indeed, we have done this. This is already published some years ago, where we reconstructed the flora biodiversity of the Atacama using fossil records in this area. And so we can go back up to 30,000 years ago and see how, you know, this ecosystem changed over time from the last glaciation event to the current interglacial period. And this study and understanding how diversity has changed over time in response to this big climate change in the glaciation, for example, can allow us to predict, or at least try to understand, what may happen in the future in response to the future climate change events that we are probably explaining. So again, so I don't have time to go into the details. We have already published this, but I think it's very important, as I mentioned, in this particular extreme environments, to really know what's there, to be able to understand how the future will affect these environments. Okay, so out of these 93 taxa and 78 plant species, we focus on 32, which we call relevant species, based on three criteria: the coverage, which is how much of the ground they cover; the persistence, which is how often are there when we go looking for them; and the distribution, which is basically how much of the altitudinal transect they cover. And this provide us with a set of 31 angiosperms. And so this Andean Atacama is dominated by angiosperms, and one gymnosperm, which is called Ephedra americana. And here in this graph, I show the location where we found these species. Is the intensity of the color indicates the persistence, how often we find them in this location. Here we have the altitude. And here you can see we have different plant families. The biggest plant families are Asteraceae and Poaceae. We have different photosynthesis types, different life forms, and about a third of these species are endemic. But as I mentioned, I think most of them, I think only Solanum chilensis has been studied a little bit, but most of them haven't been studied before at the molecular level. So here I have pictures for all the 32 species, and what we decided to do was to sequence them using Illumina parent sequencing, starting at the transcriptome, just to have a sense for what's expressed in these species. And with the idea that understanding what the genetic features of these Atacama species are, we can try to answer this question: what are the features that allow them to survive in this extreme environment. And just to give you a sense of the breadth of this project, this is a picture of one of the stations that we are using to work. And basically, we have sequence information for everything that you can see, because the other species are much less abundant or ephemeral species that you can find maybe 1 year out of the 10 years that would be in the transect. So we really have a very good coverage in terms of the biodiversity that is present most of the time. And if you ever visit these areas, basically everything that you can see out of the oasis, we have some kind of sequence information for that now. Okay, so what we have done, well, we've done several things, but I'll show you one of the projects that we published recently in collaboration with Gloria Carusi from the Center for Genomics and Systems Biology at New York University. I think two people here were important, and in particular, I'm going to talk about the work of Gil Eschel, which did some positive selection analysis to try to understand adapted features of the Atacama species. So we took these 32 Atacama species, which gave us a lot of information in terms of the total protein coding genes identified. We have over 480,000 plant protein coding genes and more than 300,000 new protein coding genes. So to try to make sense of all this information, we compared the Atacama species with 32 sister species. So these are species that are phylogenetically related to the Atacama species that don't live in Atacama. So presumably they're not adapted to the extreme conditions of the Atacama species. And we also included 6 model species just to bring annotation, which is, for example, Arabidopsis, where most of the work in terms of characterization and functional analysis have been done. We took these 70 species, the 32 species from Atacama, the 32 sister species, and the 6 reference genomes into what we call a big plant pipeline for phylogenomic analysis. And this is a complicated slide, but I will just say that in the first row here, what basically we're doing is to identify orthologs between all these species. So we have sequence clustering of the sequences using BLAST and MCL. Then we have alignment of all the sequences in the clusters using MAFT. Then we build gene family trees for all these alignments. And from these gene family trees, we can identify the orthologs for all the species. And in the second row here, basically what we did is a total evidence tree for the phylogenomic analysis. And so we concatenate the orthologs, which is what's indicated here, to build a big phylogenomic matrix. So in each of the rows, we have the species, and each of the columns, we have the characters, which in this case are protein sequences for the orthologs. And with this phylogenetic matrix, we build a total evidence tree that then is used to understand what are the unique features of the Atacama species that distinguish them from the sister species. So, for example, we can stand on this branch of the tree and ask what are the characters that distinguish an Atacama species that may be here from a sister species that would be here. And these allow us to identify protein sequences, because these are the characters that we're analyzing, and then associate these protein sequences to biological processes such as the one that are shown here as an example. And what I will tell you about today is basically our DNDS analysis, which is analysis to detect positive selection, which presumably are important for adaptation in Atacama. I don't have time to go into the details again, but this was a big job in terms of computational analysis just to resolve the trees for each of the gene families and also to calculate the total evidence tree. But once we had that, Gil was able to perform DNDS analysis, and we did 2 types of analysis: a tree-dependent DNDS modeling using CodeML and branch-type models, both species-independent adaptation and ancestral adaptation. And then we also carry out a tree-independent DNDS calculation using SNP-Gini, where we compare different groups of species, for example, Atacama species versus sister species, or as shown here in this graph, drought-adapted sister versus mesic sisters, Atacama versus mesic sisters, and so on. Again, all the details are published in this paper, but I think the main idea is out of all these analyses, we were able to identify 265 gene families that are under positive selection in Atacama plant species, which are summarized in this figure. So here at the top, we have the different plant species that we have analyzed. In red, we have the Atacama transcriptome data. In black, we have the sisters transcriptome data. And in blue, in between, you can see some of the model species, such as tomato, for example, soybean, Arabidopsis, and so on. And here we have the phylogenetic plant family. So we have a big cluster of Astraeses, a big cluster of Poaceae, and the gray squares inside this graph indicate the presence of a gene family that is under positive selection. So the first thing that you can notice probably is that there is a set of gene families that are under positive selection exclusively in the Poaceae. And there is another one here that is exclusively under selection in the Astraeae plant family. And so overall, a third of the gene families are under selection in one plant family, but 2/3, more than 60% of the plants or the gene families are under positive selection in 2 or more plant families, which I think is really interesting because it suggests we have convergent evolutionary events from plants that are coming from different evolutionary trajectories coming to the same gene families under positive selection to survive in the Atacama species. The other thing that I think is interesting and about this data is that if you look at this column here on the right, this indicates the relative level of expression of the genes in these gene families from white, has no detected expression, to dark brown or reddish color, which indicates very high level of expression. And many of these genes that are under positive selection have medium to high level of expression in these plant families under field conditions, which indicates that not only the protein sequence is under positive selection, but also these genes are highly expressed and probably also they play a role for adaptation. So just to give you a flavor for the type of information that we got from analyzing these 265 positively selected gene families, here I show you the gene ontology terms annotation. So these are descriptions of the functions that these genes families may have. And I have here the main or the parent processes that are more general, so to ease the understanding of this data. So here, for example, we have response to stimuli, and we have several gene ontology terms that are associated to some of the environmental stresses that the plants must tolerate here. So, for example, response to light stimulus, as I mentioned, this is a high radiation environment, response to salt stress, etc. And here we have a big cluster of metabolic processes. And for example, one of them, which is overrepresented, is cellular nitrogen compound biosynthetic process. So the intensity of the red here indicates there are more gene families annotated to this process. So these biological processes called our attention and indicated these plants may have some metabolic adaptations that may be relevant for survival in Atacama. So based on that, we decided to do a metabolomic analysis of these Atacama plant species. We did targeted and untargeted metabolomics. So here are some of the species that we selected. In this case, we collected samples across the transect. So this is the altitude again. And we analyzed all the metabolomes from these species in the context of the various environmental parameters that we have for these study sites. And this was published last year. And this is a work in collaboration with our French colleagues, Pierre Petriac, Dominique Roland, and Yves Guibon at the INRA Bordeaux. And just to make a long story short, I think one of the main findings from this metabolomic analysis, which I think is very interesting, is that we were able to identify 39 common metabolites that track very well the plant environment. So this is the real elevation level at which the samples were collected, and this is a predicted elevation level based on these 39 common metabolites, and we have a very strong correlation, very significant correlation that it's completely lost if we permutate this data set. And this correlation is so good that we can even predict plant elevations from samples that we collected 5 years before this. So I think this is This is cool because these common metabolites that are found in this environment belong to different plant families. And I think it's very interesting targets that we can use to extrapolate to other, for example, crop species. These metabolic changes are also associated with an ecological behavior that we observe, which is facilitation. Facilitation is how when species help each other to survive in extreme environments, such as the Atacama. And here we have one example where Atriplex, which is a relative of quinoa, can extend its altitudinal range in association with Myonopsis kamachoi, which is a cacti that you may see in this picture. So here is Atriplex growing on its own in the field. Here is Atriplex growing inside the cacti. And we have quantified this. We can show that Atriplex can extend its altitudinal range in the transit when it associates with this cacti Myonopsis kamachoi. And the adaptations in the metabolism have a lot to do with this facilitation interaction. Again, this is already published this year, so for more details, you can look at this publication. Okay, so I'm near the end. So we have identified a number of biological processes that are important based on our positive selection analysis in the underground tissues, such as functions related to enhancing nutrient use efficiency, root development, such as root hair growth, for example, optimizing water capture, and biotic interactions. I didn't have time to show you all the work we have done characterizing the microbiome associated to these species, but those also plays an important role in survival. And then we also identify a number of processes in the background organs, such as genes involved in modulating flowering time, in protecting and improving the photosynthesis capacity, and also to deal with the high radiation that the plants experience in this environment. I hope I give you a sense of how we're using big data sets, integrative bioinformatics, in this case, phylogenomic models, to develop new hypotheses about how plants in Atacama survive. And we hope certainly that this can help us enable or make a greener future. So with that, I'll finish. So I have to thank again Gil Eshel, Viviana Raus, that I didn't mention before, and Thomas Dussarat, an outstanding grad student that did most of the metabolomics work. Some of the funding involved. This is a recent picture of my group, and these are some pictures that we took in the different field trips that we've been doing over the years. This is from this year. And just the last thing I'll say, I have to thank also Claudia Torre, a great friend and colleague here in the Faculty of Biological Sciences. He's a desert expert, and without him, this project would not have been possible. So this has been a true interdisciplinary collaboration with Claudio to address, and I already mentioned, to address the survival in Atacama. And these are some of the colleagues that are, some of which I mentioned related to the phylogenomic work. With that, I'll finish, and I'll be happy to take questions if there are, of course. Thank you for your attention.
Great. Thank you so much, Rodrigo. That was really interesting. We do have a fair amount of questions in the Q&A box. So I guess with that, I'll pass this over to Isabel for this next portion.
Okay. We have several questions in the Q&A. I would like to remember the attendees that you can vote for your favorite questions and not just posting them. So let's start with the first one that came in. This one is for Rodrigo. So Marwa Halmi asked, I need more elaboration on what you mean by persistence, and is it different than frequency?
Yeah, persistence is just the fact that when you go out in the field, when you do botanical surveys, you find these species, of how often you find the species when you go out in the field. That's what's defined as persistency. Yeah, I guess that's the answer to that question.
Mm-hmm. So it's for what I understand, it's the same as frequency?
No, it's not the same as frequency. So frequency, I guess, would be more related to coverage, which has to do with how how much of these species you find in the field. But I guess the easier way to understand persistent is, for example, a perennial species is probably very persistent because it will be there for many years. So every time you go, you'll find this species. But an annual species may be not as persistent because it may only germinate and flower in certain years where the climate, so for example, with the El Nino climatic event versus other. But some annuals are actually quite persistent, so that we find them every time we go. And others are not so much. And even some perennials, sometimes we cannot find them just because they're not very abundant. And they will germinate, grow, but they may die in response to certain. So persistence gives you an idea how stable, you know, the ecosystem is, and what are the main species or the most important species that define this ecosystem. The frequency, to me, at least, I don't know if this is what Marwa means, but it has to do with how much of these species is found in a given location. For example, in the picture that I have in the background. So some of them are very frequent, right? Like these jarabas, maybe that you see, these grasses that you see, but others may not be as much.
Thank you. Now it's very clear.
Okay.
So do you want to, Liana, do you want to choose the next one?
Yeah, can do. So we have a question from Dinakaran Elango, who asks, are there any efforts to understand the soil physiochemical properties, including microbes, to understand the adaptation of plants in different altitudes of the environment? So this would be to Rodrigo as well.
Yeah, I mean, I didn't have time to go into too much detail, but yes, we have analyzed the microbiome associated to these species across the entire transect, both in bare soil and as well as in the rhizospheres, which is the soil associated to the roots. So we do have a fairly good understanding of how these microbial communities change from bare soil to the root, and also how they change over altitude. And we have identified some taxa, so the microbial taxa in this case, that may contribute functions for survival. So for example, we have an enrichment of nitrogen-fixing microorganisms associated to the roots of these plants, which makes sense because this is, as I mentioned, it's a very low nitrogen environment. So these plants probably recruit from the soil bacteria that are able to fix nitrogen from the air, and this provides with the nitrogen they do. And we have done, as I breathed through it, but we have done these studies over many years. So we have now 13 years going to the field, and every year we measure biodiversity, we measure persistent coverage, and all of these parameters. So this is also in response to the next question, which is related. So I threw it out there as well.
Okay, great. Thank you so much. I want to kind of pivot and ask, in the similar vein to Claudia, if with your salinity and drought work as well, if microbes are something that your lab or your collaborators are taking into consideration when thinking about tolerance to these kinds of stresses. So this isn't directly in the chat box, but that then got me interested in other applications as well for that kind of work.
Sorry, I didn't hear it very well.
Sorry. So my question, to build off of the previous question, is if your lab or your collaborators have looked at microbes and also the physiochemical properties with soil as it relates to salinity stress and drought stress in your work in tomato and kiwi. I'm just curious if that's something that you all have looked at.
Yeah, the other aim of the project is looking for promoting rhizobacteria that could help plants to overcome salt and growth stress. Yeah, and some of them were taken from the tomatoes grown in the Atacama Desert, and
okay.
we are at present we are evaluating if they confer some tolerance in plants that are grown for commercial aims.
Okay, interesting. Thank you. So I guess going back to the chat box here, so this is also related to previous question, or rather also for Rodrigo. So Sadar asked, what morphophysiological data was collected for the Atacama species?
What? Sorry, what
morphophysiological data. So I guess related to morphology, different physiological indicators.
I mean, we have collected sort of the above ground morphophysiological data for, I think, most of the species. For the below ground is harder, and we have chosen to focus on a couple of species, and one in particular, which I think is very cool. It's called Homphansechia doelis, a fabaceae that produces a edible tuberous root. And this is one example of the ones that I mentioned in the introduction, that these plants have intrinsic value because they were food for the local communities that live here. And so this species, one of the adaptations it has is that produces these tuber roots that allow persistence over the years, right? So when it rains, which it does rain in the desert, not very often, but it does, then this tuber germinates and provides the next generation. But if not, then it remains as this potato basically in the ground, or I don't know yet for how many years. So this is one interesting example of how this plant escapes or survives the most extreme part of the year when there is absolutely no water. But as I mentioned, also, I mean, a lot of these species haven't studied before, and you know, we have done a lot of work, but still there's many questions that remains to be answered, and so we have to focus on some of them to be able to advance. Yeah, I'm not sure it answers the question, but yes, it's the information we have. Yeah.
Okay, thank you. And I guess if there's any follow-up is needed, then that can be typed into the chat box as well. So Dina Arampali has a question I think is somewhat related to this. And she asks, I may have missed it. What was the temperature in the Atacama Desert? It's interesting to see that species from the Asteraceae family are most abundant. Is anyone working on the adaptation at the gene level of these species to desert environments?
Right. So this is a temperate desert because it's high up in the mountains, so it's not so hot, but there is also a temperature, as I said, temperature climb as you go up in the mountain. So it's hotter at the bottom of the mountain, and I would say in the summer the average would be probably around 30 Celsius degrees, and at the top of the mountain the average may be close to 20, between 15 and 20 Celsius degrees. This is more or less it's not as hot as, let's say, the Death Valley in the United States, right? But this is because we're high in the mountains. Yeah, I guess that's the question. Yeah, but we have meteorological data. If anybody is interested, we can certainly provide that, and we have recordings every hour for the last 8 years, I think, worth of data in terms of temperature, precipitation, humidity, and radiation, and other parameters. So we're happy to share that data if anybody's interested.
Very interesting, yeah. So we have a few other questions. I'm just going to group together that are going back to this question of persistence. So Sadar asked how many seasons the species were checked for persistence.
Right. So we do this the surveys, we do them every year since 2010 at the end of the rainy season. So in the Atacama Desert we have rains in February. Because of the effect of the Amazon evaporation that hits the Andes Mountain on the other side, then you have condensation and there is some precipitation. So this happens in the southern hemisphere in February most of the time. So we go there in March after the rainy season, because this is where you would find most of the species because of the precipitation. So mostly annuals, because the perennials are going to be there, but the annual species you only find when there is some water. We have done some analysis of the diversity in other seasons, like in winter and fall, but not as regular. And what we find is actually the same species minus the annual. So I think this is the reason why we always go, as I said, at the end of the rainy season in this summer.
Okay, Thanks. That's really interesting. And so this question is also related to that. So when you go back in March to do these measurements, do you have permanent plots, or is it just you are walking through this ecosystem and?
So we have both in a way, because these are pristine environments. This is not a like a university station or something. This is out in the field, but we have them GPS tagged. So we will come back always to the same locations. But it's not something that we can really call an experimental station, because, as I said, it's just public land, it's open, but it's just a pristine environment.
Okay, thank you. And now I have two questions related to phylogeny, and then I have some questions that I'd like to ask both of you that are a bit more general. But the first one is if you could share some insights on the software and the kinds of tools that you use for constructing these phylogenetic trees. And also, if you could maybe speak to the importance of these phylogenetic studies to understand climate change. So whatever order you'd like to take that in.
So I guess the second one first. I think it's important in the sense that by comparing species that are highly adapted to a very drought environment, for example, just thinking about the lack of water in Atacama, it could give you a sense for mechanisms that could be useful to improve crops, such as, some of the candidates that Claudia was talking about, right? She took them from the literature, but one could take some of the genes that we identify in Solanum chilense, for example, and other solanaceae, and I think there would be probably a higher chance. I dare to say that there will be a higher chance that those might work in a commercial variety, just because these genes are important for surviving in a very extreme environment such as Atacama, and already is in the same plant family, and also may be conserved in other plant families. So this is, I think, the importance of this phylogenetic study, because it just provides you candidates. And we're doing some follow-up studies again. So this is a big data set, so we cannot study all of them, but we have selected some of the genes that have annotation, and we're doing follow-up studies in some plants of commercial interest, such as maize or rice. And then we're also studying some genes that have no annotation, which I think is very interesting as well, because we have no annotation about them, we know nothing about their function. They might give us also new mechanisms that are important for adaptation. So I think this is the main relevance of this phylogenetic study in terms of how we face climate change. And of course, I forgot the first one, which was related about the tools, the software. So the tools, I briefly mentioned them when I talked about the phylogenetic pipeline. So for the phylogenetic tree construction, we use standard tools such as BLAST, MAFT, and TNT. And then the same basically for the big phylogenetic tree with complete phylogeny, using just a bigger phylogenetic matrix. And then for DNDS analysis, which is to select the positive selection, we use SNP-Genie and CodeML. Those are the two software tools that we use to do the positive selection analysis.
Yeah, thank you so much. And I think also speaking to your work on phylogeny, and that you didn't quite mention, but I think is also really important. It's also, all this is a record when we think about taxonomy as to what's been present and how things are changing moving forward. So I think that's also something that has a lot of value when we think of these studies as well. I have a general question for the both of you. Both of you are involved in really large collaborations with a lot of institutions from around the world. I was curious if you both could speak to how these collaborations came about, how people who work in this kind of field can also get involved, and how you just manage these. I know that science is becoming more and more interdisciplinary, and both of these seem like really great examples of that. So how do you guys work towards that?
You want to tackle that, Claudia?
Go ahead.
So, I mean, this is the project that I talked about. I mean, it's been very many years, but it started really slow. So at the beginning, it was mostly exploratory, going with Claudio into the field, observing the diversity that was there. And slowly we started to build a foundational data set in terms of, as I mentioned, climate, the soil features that I didn't talk about, but we've done in-depth analysis of the soil to understand the physical characteristic as well as the basically biochemical properties of the soil. And so over the years, as we were building these data sets, it attracted attention from colleagues. And so for specific questions, so with Gloria, we collaborated on the phylogenetic analysis. Then once we had the phylogenetic analysis done, we built a collaboration with the French group to do the metabolomics. People joined from Chile also to study the microbiome associated. So in a way, I mean, over more than a decade, these collaborations have been building slowly, just to address specific questions that were derived from. And we have more, I think, ongoing projects. We're trying to set up a project with the European Space Agency, for example, to try to test some of these Atacama species in the International Space Station to see whether they can cope better with the stresses of microgravity, for example, than other related species. So I don't know, maybe I'm rambling a little bit, but the idea is that, you know, as we build more and more data, this attracts more colleagues with specific ideas, and we basically take advantage of that and move forward.
Great, thank you.
Yeah, it's basically what Rodrigo mentioned, no? Is the access to diverse expertise, accelerate the progress of the results. You have the availability to meet, to join efforts, to have access to more funding, to apply to other fundings, and so on.
Oh, we lost Claudia.
We lost Claudia for a moment.
Okay, I guess let's take a moment to see if she reconnects. But yes, if anyone in the audience has any other questions, Rodrigo, I feel a bit bad because I feel you're getting bombarded right now, but I have a personal, like a question as it relates to my research. So I really work with photosynthesis and fluctuating light environments and the role of light stress in plant productivity. I was curious, and you mentioned this on one of your last slides related to Arabidopsis orthologs, but I was curious if there's any clues as to how these plants cope with high light stress in this already really harsh environment and what they're doing to deal with that.
Yeah, I mean, that's a great question. I mean, we have some ideas in the sense that we have identified some gene families that may be directly involved in dealing with the high stress, both in terms of the chloroplast machinery, as well as the redox stress associated with the high radiation. But that said, we haven't done any in-depth following up on that. So if you are interested, I'll be happy to chat about that, because I think this is something that I've discussed with some colleagues that it may be very interesting. Also thinking about, I didn't talk about this, but as you go up in the mountains, of course, the pressure of CO2 and oxygen changes. So these may also. So I'd be happy to talk about that in more detail if you're interested.
Yeah, I would love that. I have a bunch of other questions, but I'll defer to others in the chat. Claudia, so glad that you're able to reconnect. I don't know if you'd like to continue on what you're speaking about related to collaboration.
Yeah, it was the same idea. So you have more opportunity to apply to more fundings and so on, and to share different experiences, to hire new students or professionals, to guide them, and so on. So it's all the same idea. Yeah.
Okay, thank you. Let's see. Great. Isabel, do you have any questions, or does anyone else have any questions from the audience?
Oh, there is another question from Dina Rampali. I think, Rodrigo, you are already answering.
I was trying to answer some of the questions in the chat.
Yes.
So you found the species
So
from Australia? Yeah.
I'm going to
Sorry.
read out loud so that the people from
Go ahead.
YouTube really know what we are saying. So the question is: you found the species from Asteraceae family is most abundant in Atacama Desert. Do you want to elaborate?
Yeah, so what I said is that we have more species from the Poaceae family in terms of all the species that are most important based on our criteria. So out of the 32, the Asteraceae is a very prominent plant family in the Atacama. And it's specifically so the picture that I have in my background is from one of the high stations. I think this was taken at about 4,300 meters above sea level. And basically all that yellowish thing you see in the background, these are all perennial grasses. And so this is where they're most dominant. But there's also other species that are in some of the other parts of the transect, not necessarily the most abundant, so that with the more coverage. But again, so what I mentioned related to Asteraceae family is that we have more species of Poaceae than other plant families in here. I think that should answer that question.
Great. Well, I guess, Isabel, if we don't have any questions, I think that we can go ahead and start wrapping up.
I just have one last question. It's for Claudia. So if you I always fascinated by how the genetic tools can speed up the development of new varieties. And I wanted to ask you if you can give an estimation of the comparison of the time when the starting point would be when in any other research is found a candidate gene, like for example the one that has been presented by Rodrigo, from that starting point until you develop a line that could be planted on the field, how much time could be there in between, and comparison using these genetic tools and using the conventional breeding program?
Yeah, it's a very good question. And that's the reason why we are moving to these technologies, because the traditional technologies by crossing plants can take around 15 years until you have the new variety that is able to be commercialized and you have the variety in the field, as you mentioned. And by the editing strategies, you may take a half time, as we calculate. In the case of producing transgenic plants, it can take the same time than the traditional methods, because these plants have to follow strict regulations that takes a lot of time. So when we compare the traditional methods with the transgenic methods, it's almost the same. But when you choose to edit the plants, it can take half time.
Great, that it's really an improvement.
Yeah, of course. Of course, we have to move fast in this scenario.
So, Liana, I think we can just start to wrap up.
Well, first of all, I want to say thank you so much to both Claudia and Rodrigo for your presentations and your insights. They're really valuable. They're really interesting. And I'm personally glad that we were able to listen to them. With that, I'd like to mention that in the next week, we'll have 2 more webinars related to this plant science-based solutions for climate change. So on Monday, we'll have a talk by Lisa Ainsworth of the University of Illinois and Amanda Kavanaugh from the University of Essex. And on Wednesday, we'll have a talk from Mark Tester from KAUST in Saudi Arabia and Robin Allen from CSIRO in Australia. So if you'd like to learn more about these different initiatives and work for plant sciences working towards solutions for climate change, please be sure to attend. Isabel, if you have anything else that you'd like to mention, but we just want to say thank you so much for attending.
Just the same as you. Thank you so much for attending, and it was great to have a co-host this time, Liana. So thank you so much, and thank you, Claudia and Rodrigo, for devoting your time to this webinar and explaining what you do in your research, and everyone who attended and stay until the very end. Thank you so much.
Thank
Thank
you.
you. Bye.